Today we have assembled to discuss about pleiotrophy. Retired university professor in botany from Ranchi Women's College, Ranchi. Now, what do we mean by the term pleiotrophy? Single gene affects more than one trait. Pleiotrophy is the effect of a single gene on more than one trait or characteristic. Pleiotropy is derived from the Greek words pleio, which means many, and trophic, which means affecting. Genes that affect multiple, apparently unrelated phenotypes are thus called pleiotropic genes. In this diagram, we can see that gene X can control trait 1, trait 2, and trait 3. A pleiotropic gene is a single gene that controls more than one trait. Pleiotropy should not be confused with polygenic traits in which multiple alleles are responsible for a single trait or multiple genes are responsible for single trait or multiple alleles in which a gene may have more than two alleles. Pleiotropy refers to the fact that most proteins have multiple roles in distinct cell types. Any genetic change that alters gene expression or function can potentially hide wide-ranging effects in a variety of tissues. Pleiotropy describes the genetic effect of a single gene on multiple phenotypic traits. Pleiotropy occurs when a gene product interacts with multiple other proteins or catalyzes multiple reactions. Now we will see it with the help of some examples. The most common example can be seen in pea, pleiotropy in pea. Gregor John Mendel, while studying inheritance in pea plants, made several interesting observations regarding the color of various plant components. Mendel noticed that plants with colored seed coats always had colored flowers and colored leaf axils. He also observed that pea plants with colorless seed coats always had white flowers and no pigmentation on their axil. Now it is well known that Mendel's observation were the result of pleiotropy, or the phenomenon in which a single gene contributes to multiple phenotypic traits. In this case, seed coat color gene, denoted small a, was not only responsible for seed coat color, but also for flower and axil pigmentation. The other example of pleiotropy can be seen in Drosophila, vestigial gene Vg of fruit fly. In Drosophila, vestigial gene plays a crucial role in wing development. Wing development, no doubt, is done by this Vg, but along with that, it also controls position of bristles on a fly's scutellum. You can see scutellum here, and number of eggs string in flies ovaries, and also length of flies life, कितना लंबा life होगा. Example of pleiotropy in cat, pigmentation and deafness in cats. Hathi and Jones, 2005, while studying fur color and deafness in cats, observed that approximately 40% cats with white fur and blue eyes are deaf. White fur and blue eyes are deaf. It was also found that white cats with one blue eye and one yellow-orange eye were deaf only on the blue-eyed side. Frizzle feather trait in chicken. Chicken exhibit various traits affected by pleiotropic genes. Some chickens exhibit frizzle feather trait where their feathers curl outwards and upwards rather than lying flat against the body. Frizzle gene exhibits pleiotropic nature as along with producing defective feathers, it also causes abnormal body temperature, high metabolic and blood flow rates, greater digestive capacity, accelerated heart rate, and delayed sexual maturity. Females who have this allele also lay fewer eggs than their wild type counterparts. Comb mass and physiological structures related with reproductive abilities are also closely linked. Both males and females with larger combs have higher bone density and strength, which allows females to deposit more calcium into egg cells. Frizzle feather are found to stem from a deletion in the genomic region coding for alpha carotene. Deletion means loss. Now this slide shows frizzle feather trait in chicken. You can see the feathers are moving, bending towards the body, not away from the body, but towards the body. Now there are certain examples of pleiotropy in human. Phenyl Ketone Urea (PKU), one of the most widely cited examples of pleiotropy in human is PKU. This order is caused by a deficiency of the enzyme phenylalanine hydrolase, which is necessary to convert the essential amino acid phenylalanine to tyrosine. A defect in the single gene that codes for this enzyme therefore results in the multiple phenotypes associated with PKU, including mental retardation, eczema, reduced hair and skin pigmentation, defects that make affected individual lighter skin, as reported by Paul 2000. It is caused by large number of mutations in the single gene on chromosome number 12 that codes for enzyme phenylalanine hydroxylase, which converts amino acid phenylalanine to tyrosine. Because the enzyme is not found, level of phenylalanine increases, which creates toxicity and can become fatal. Second example of pleiotropy in human is sickle cell anemia. Sickle cell anemia is a human disease originating in warm lowland tropical areas where malaria is common. Sickle celled individuals suffer from a number of problems, all of which are pleiotropic effects of sickle cell allele. Sickle cell allele has incorrection in the sequence of amino acids. In place of glutamic acid, valine is introduced. Sickle cell anemia is an example of a pleiotropic human blood disease. It is caused by an allele that incorrectly codes for hemoglobin, causing normally round red blood cells to become sickle cell. As a result, the cells are inflexible and cannot easily flow through blood vessels, increasing the risk of blood clots and possibly depriving vital organs of oxygen by reducing efficiency of RBC, red blood corpuscles, in carrying oxygen. Some complications associated with sickle cell anemia include pain, damaged organs, strokes, high blood pressure, loss of vision, fatigue, and delayed growth. Sickle red blood cells also have shortened lifespan and die prematurely. On the contrary, individuals who are carriers of sickle cell disease with one sickle cell gene and one normal hemoglobin gene have some protective advantage against malaria. As a result, the frequencies of sickle cell carriers are high in malaria endemic areas. Now, this diagram shows these are the normal RBCs and this one is the sickle shaped RBC. This is efficient in carrying oxygen and this sickle shaped cannot carry oxygen efficiently. Third example of pleiotropy in human is Marfan syndrome. Marfan syndrome is an order in human in which one gene is responsible for a number of symptoms, including thinness, joint hypermobility, limb elongation, lens dislocation, and increased susceptibility to heart disease. Marfan syndrome (MPS) is an autosomal dominant disorder which arises from mutation in FBINI gene which codes for glycoprotein fibrillin 1, a major constituent of extracellular microfibrils which form connective tissue. Because these fibers are found in tissue throughout the body, mutation in this gene can have a widespread effect on certain systems including cardiovascular, skeletal and nervous system as well as the eyes and lungs. Mutation of all the genes NVN that code for DNA damage repair protein NVS1 leads to microcephaly, immunodeficiency and cancer predisposition in Niemann-Pick syndrome, which has an autosomal recessive pattern of inheritance. Fourth example of pleiotropy in human is Holt-Oram syndrome (HOS). Mutation in the gene that codes for transcription factor TBX5 cause the cardiac and limb defects called HOS. HOS is an autosomal dominant condition characterized by congenital heart defects, tracheal and renal anomalies, thumb anomaly, arterial septal defects, and deafness are also associated with this syndrome. The T-box transcription factor TBX5 is a protein that in human is coded by the TBX5 gene. This gene is located on the long arm of chromosome 12. The TBX5 gene affects development of forelimb and heart by triggering fibroblast growth factor FGF10. Antagonistic pleiotrophy. Sometimes a pleiotropic gene may be both harmful and beneficial to an organism, which is referred to as antagonistic pleiotropy. Williams 1957 proposed antagonistic pleiotropy hypothesis. He suggested that some genes responsible for increased fitness in the younger fertile organism contribute to decreased fitness later in life, which may give an evolutionary explanation for senescence. An example is the P53 gene in human, which suppresses cancer but also suppresses stem cells, which allow the body to renew and replace deteriorating tissue during aging. It also prevents cells with DNA damage from dividing. This situation is therefore an example of antagonistic pleiotropy in which the expression of a single gene causes competing effects, some of which are beneficial and some of which are detrimental to the fitness of an organism. Now, with this, I think I have been able to tell about the pleiotropy, and in the next meeting, we will discuss polygenic gene inheritance, and we would now end the class. The class is being ended.